Okay. Okay, we're back to this now. And let me plug in my tablet so we can start drawing again after our brief intermission. So we have the cell membrane, the nucleus, all its parts, the rough ER, the smooth ER, the Golgi apparatus, vesicles, and something we don't have, a little bit larger than the vesicles, a vacuole. And that would be number 8. Let's change color. Let's see. About the same size. And that would be the lysosome. Okay, so we have number 8 vesicle, number 9, the lysosome. Then we have a region that is near the nucleus, a denser region. There's no membrane, it's just a denser region, and inside the centrome there is also little fibers. Let's draw them out here. Something like this. They're all in an area of this. We call this whole area there, this whole area we call centrosome. But these two things there, which are a bunch of microtubules of fibers there, are the centrioles that only animals have. And that's color. And our last thing is you have all over, you have different parts. We have the cytoskeleton. And yes, there's 3 different parts, 3 different types. You do not have to draw it, the 3 different parts. Okay, there we have all the organelles, and they're labeled for everything. So next what we want to do is have another slide and start writing down what those functions are for those different organs. So our first one, you could write cell membrane again here in case you didn't remember. Controls what enters and exits the cell. So you could say molecules or substances. And then 2 was the nucleus. Controls all cellular activity and contains all of cell's genetic material. Now give me a second to check and see what's A, B, C, D, and E, and I'll write them for you. A, okay. A was a nucleolus, so manufactures ribosomal subunits, or you can say parts of the of the ribosome. And B was the nuclear membrane, protects the DNA and all other material in the nucleus. C was the chromosome and is the DNA. Yes, it's a little more, but we're not in the DNA. D was the nuclear pores, controls what enters and exits the nucleus. And E was a chromatin, so once again, it is the DNA. Okay, now we're at 3. 3 was the mitochondria, site of cellular respiration. Cellular respiration. Or if you said the cell's power plant, it would be fine. It says what it does. 4 was the ER endoplasmic reticulum. And let me stop to make sure which one was 4A, which one was 4A. Okay, 4A. Oh, that's not the color I want. A was a rough ER, and you could write site of ribosomes, transports and modifies proteins. B, smooth ER, transports proteins, but it also makes lipids, which include hormones and detoxifies alcohol and drugs, and that includes any other poison and other poisons too. That's what alcohol and drugs are. They are considered toxins to the body. Now 5, let's take another color for 5. Let's take this green. 5 is the ribosomes. So you can simply write site. Site means where it takes place. Site of protein synthesis means making proteins. And some of you have been saying where amino acids are linked, forming proteins. So people are saying it different ways, but all of them are correct. Six is the Golgi apparatus. Let's take another color again. And processes and packages proteins for shipping or transporting out of the cell. Either packaging and processing the proteins for shipping or for transporting it out of the cell. Now we have 7. Oops, I need our pen. Now what color? 7. 7 is the vesicle. So it is a membrane bound sac for transporting proteins and other products and other substances, say like lipids. Say a hormone is a lipid, it still has to be transported. It could be transporting it from the endoplasmic reticulum to the Golgi apparatus. It could be transporting it from the Golgi apparatus to the plasma membrane or cell membrane, where then it's then released through exocytosis. Then we have 8 vacuoles. These are membrane-bound sacs for storage of water, food, etc. Number 9 is the lysosome, but we forgot to put a number for our 2 last ones, so let's number them. Go back. Let's put number 10 for the centrosome and centrioles, do them together, and number 11 for the cytoskeleton. Okay, so number 9 is the lysosome. And once again, membrane-bound sac containing enzymes for breaking down food damaged organelles, and / or bacteria. Now the damaged organelles, that means it breaks them down into components that can be reused again. Now we're to number 10. And number 10 was the centrosomes and the centrioles. And so I'm going to write centrosome here first. Dense region of the cytoplasm where the cytoskeleton or microtubules are manufactured. Including microtubules used for dividing the DNA during cell division. Now that's a lot, but if you just get it, it makes microtubules, you're fine, because we'll go back to this when we go through cell division. And then centrioles, they are connected, because we don't know everything well, but we know they're connected with making cilia and flagella. We don't have flagella, but we have cilia in our trachea or in our lungs, and people who have a genetic defect that where they don't have the centrioles, they don't have cilia in their trachea because they can't make them. And here comes our last one. And that's 11. And there, you know that the cytoskeleton is divided into 3 parts. So here you write microtubules, they help to maintain the shape of the cell and move around the organelles. Intermediate filaments anchor the organelles. They help keep the organelles in place. Anchor the organelles. And the last one is the micro filaments, they can change the shape of the cell slightly when necessary, are responsible for movement such as using your muscles. And yes, you can say it different ways. But if you draw and label like I've done there, and you see my drawings are fairly simple, and you write what I wrote, and some of it I wrote more than you need, but I know I wrote it in different ways. You don't have to say it exactly what I said, but you're going to get all of the points. See you at the test.